let's review the basics of brewing beer, remembering that the few simple steps that I've outlined for you here on the left really don't capture the complexity and nuance that make brewing beer an art form. Let's remember too that brewing beer is a conversion-based process whereby the sugars that are stored as starch polymers have to be mobilized as simple sugars for the yeast to ferment them to ethanol. That process begins with malting the grain, and here's a picture of a malt house where we have a temperature and humidity controlled environment that causes the seeds to begin to germinate. The germination of those seeds allows the development of grain amylases. These are enzymes that are responsible for the conversion of starch to fermentable sugars. So it's actually the grain itself that's the source of the machinery that's used to degrade the starch into those simple sugars. And once we've germinated, then we heat up the grains and slow down that process once again, dry them down so that they're good for storage. Now, once we're ready to do our brewing, then we grind the grain and extract with hot water, and this process is referred to as the mash in. Here on the right, we show a picture of someone stirring in the grain into a vat of water. They're dissolving the starches. The amylases are acting on them and converting them to simple sugars. At this stage, we're ready for lautering or filtering. We remove the solid material from the grain. That's the lautering step. And then we sparge or rinse. If we want to extract more sugar from the grain, then we'll rinse it with fresh, clean hot water and remove a little bit more sugar from the grain. At this point, we're ready to boil the wort. So now we'll refer to that saccharine liquid as wort. We're ready to boil. Why? Well, reason number one is to sterilize. We want to be able to control what the fermenter is that's fermenting our simple sugars. Not only that, we want to control the sugar profile, what I'll call the sugar profile. That is to say, what sugars are available for fermentation and what sugars are not. And the way this works is by denaturation of those same amylases. We've talked about protein denaturation before. That is the process where we kill the protein. In this case, we apply heat, that is boiling, and that unfolds the structure of those amylases that's responsible for their ability to degrade the sugar or degrade the starch. And that means that they're no longer going to be able to change whatever the carbohydrate profile is, right? So whatever starch we've already turned into simple sugars, that's all we're going to get. So we can lock in a sugar profile by denaturing those amylases. Then we chill things down right away so that we can then add our yeast. And this is just an immersion cooler. There's a couple of ways to do this. You can imagine attaching these coils to cold water inlet, and we'll immerse this in our wort and rapidly cool our wort that way. But a little more efficient way to do that is to pump the hot wort through the coils that are immersed in a cold water bath. And that way we'll quickly bring the thing back down to the temperature where we want to do our fermentation, and then we pitch the yeast. And in this picture we're showing, there's another home brewing photograph where we have activated our yeast, and that's shown in this little Pyrex dish here. And then our active yeast we're pouring into our fermentation vessel, and that's pitching the yeast. And now we're controlling exactly what fermenters are available there. No wild fermenters, please, unless that's the goal. So, and then finally there's the fermentation step where we pop an airlock or fermentation lock onto our fermentation vessel, and that prevents unwanted bacteria from migrating into our fermentation or unwanted yeast for that matter, to migrating into our fermentation, and it also allows the evolution of CO2, right? The CO2 bubbles out into the atmosphere now. The steps that I'm not showing here after fermentation, I don't want to dwell on, but let's simply say that we have to rack, right? And no matter what we're going to do next, we're going to rack off the liquid and try to separate as much as possible fine solid material from the liquid. And then we're either going to keg or bottle. And there's two different things that happen here, each of which result in carbonation. In the keg, we actually charge it with pressurized carbon dioxide. But if we're going to put it into a bottle, then we prime. And this is an interesting step whereby we add sugar, more sugar. So you say, well, why? We just fermented all of our sugar. Why are we adding it back? Well, we're going to go through another fermentation phase, just a little bit of fermentation after we seal those bottles. So the sugar is going to allow the fermentation to proceed after we've bottled, and that's going to allow CO2 to evolve again, but in this case, it's not going to escape. It's going to remain in the bottle and carbonate our beverage. Those are the basic steps. Again, simplified, right? But that's the fundamental steps that take us from grain to our fermented beverage. I want to belabor this a little bit more by showing you a cartoon, and let's just say that the mash-in is a complicated process, and what I really want you to get a sense of here is the number of amylases that are involved, the complexity of the process, and the complexity of the sugars that you can generate during the mash-in. Here's a starch polymer. In plants, there are two starch polymers. One is unbranched, that is amylose, and one is branched, that is amylopectin. Here we see a branch point. It's important because specific amylases are required to cleave this kind of branch, different amylases than are required to cleave this kind of branch. Let's not belabor it too much, just to say that the polymer is built of glucose, right? But the number of complex sugars that we can generate from this polymer is pretty varied. Let's just look at a couple of possibilities. Here's something called a limit dextrin. This is 3 carbohydrates all linked together, and there's a branch point there, right? So that's going to be something that's a complicated structure. Here's another dimer or disaccharide, maltose, right? A dimer of glucose. So basically, again, I don't want to go too deeply into this for you unless if you're really interested, go read this paper. But bottom line is that one enzyme can cleave one kind of bond. Alpha glucosidase is going to cleave this sugar from this end. Alpha amylase is going to cleave here at this alpha 1 to 6 linkage, right, limiting this limit dextrin. But there's another enzyme, limit dextrinase, that can cleave that thing up into different pieces. And ultimately, all of these enzymes together are working towards giving us as much glucose as possible that's ready for the yeast to ferment. So different amylases are active at different temperatures. The only point there is that we can control the temperature, and therefore we can control, in principle, exactly what kind of carbohydrate profile we generate. How do we do that? We do that by varying the mash-in temperature. And we got 2 possible goals and a bunch of goals that might be in between there. One is to maximize the amount of conversion to glucose, right? We're going to generate as much sugar as possible. We're going to generate as much fuel for the yeast as possible, and we'll generate as much ethanol as possible. The other end of the spectrum would be to maximize the amount of complex sugars that are available. And that might be accomplished in a variety of ways, but one possible way might be deciding exactly which enzymes are available or unavailable during the mash-in. For example, if we kill limit dextrinase by denaturing it perhaps, then we're never going to be able to break this kind of molecule down, and we're never going to be able to use those to ferment into ethanol. These going to add, well, they're going to add body, right? They're going to add density. They're also going to add flavor. Okay, so we see where the flavor is coming from here by considering what happens during the mash-in with these amylases. Okay,